Welcome to Cow Talks. I'm Chris Pravat, Beef Cattle and Forage Commodity Director for the Alabama Farmers Federation.
And I am Marcelo Valau, Forage Extension Specialist with the University of Florida.
And this is our podcast, where we dive deep into the main topics affecting livestock and forage production in the southeastern United States.
From the mainstream media to new technologies straight from our research stations.
From cattle prices to international trade.
From our pastures and beyond.
Join us on this journey as we tackle the main issues affecting our producers and the sustainability of our production systems.
Welcome to Cow Talks. This is Marcelo Valau, Forage Extension Specialist with the University of Florida, and your host. And today co-hosting with me is my PhD student, Miranda Emery. She is also working with the Cervity Health Research Initiative, and that's why we have our special guest today. Dr. John Lednicky is a research professor in environmental and global health here at UF with his background in microbiology and virology. So his current research focuses in areas of virus discovery, virus surveillance, and influenza virus studies, which is a topic we have been discussing here in this podcast recently with the H5N1. John, welcome to our podcast.
Well, thank you for inviting me. Excited to be here.
Awesome.
Thank you, Marcelo. I'm really glad to be here and excited for today's talk.
And I appreciate your help. So let's, we recently interviewed Dr. Ben Anderson also with the Environmental and Global Health, and he briefly mentioned about the concept of One Health. And I would like to take the opportunity here to expand. What is One Health?
That's a very good question. And a lot of people have their own explanations for One Health. The way I explain it to my students is when we look at the well-being of animals and humans and actually our environment, everything's interconnected. And we often have a human-centric viewpoint. But I'd like to remind people that when it comes to emerging diseases, most of them come from animals. And some of the most common or the better known ideas are HIV, which came from primates that jumped into humans. And then we have Ebola virus, Mpox, and so forth and so on. Influenza viruses also tend to come from birds if they're influenza A viruses. So it turns out we also transmit diseases to animals. And there are a lot of emerging diseases now. And one reason for that is because we're degrading, our forests and that sort of thing. And as we start doing that, a lot of the pathogens end up in new hosts. So in One Health, it's an approach to the health of the planet by looking at what's going on in humans, animals, and the environment, and understanding that everything is interconnected.
Perfect. It's a big system, and we're working in systems.
Yes.
It's so interesting, this concept of One Health, because we tend to work in only sections of the systems and we don't integrate that. And I love the concept of integrating all of that knowledge, which is a lot of the work we do with the Cervity Health Initiative, right?
Yes, that's correct.
Yeah, and speaking of that, we heard a lot about the One Health approach that recognizes the health of people that is closely related and connected to the health of animals and our shared environment. Can you share any success stories where a One Health strategy worked well?
Well, in our work with deer, farm deer, it turns out deer have receptors for SARS coronavirus 2, the virus that causes COVID. And there was concern that all the deer that are in contact or close contact with humans would die. Now, here at the University of Florida, we're looking at farm deer. So the deer in pens are not going to be in close contact with wild animals. But what's very interesting is that the human caretakers can transmit their viruses to the deer. So by recognizing that and interrupting that transmission chain, we were able to show that in Florida, at least with the farm deer, there was hardly any incidence of transmission of the virus from humans to the deer. And we also learned in other studies looking at wild deer that the incidence was very low in Florida compared to other states. Why would that be the case? We don't really fully understand, but one of the things that we do very well here at Cherry is we confirm what the animals have by doing molecular tests and microbiology tests. Whereas other people don't do that. They make a clinical diagnosis by just looking at the animal and second guessing what the animal has. Here we've been able to really contain the spread of that particular virus to farmed animals. And I think that's been very good for the farming community here, those people doing deer farming, because all the concerns that people had have gone away. Right? It turns out we're not causing a catastrophe. We didn't create a situation where all the deer got sick and then all the people surrounding the deer died or anything like that, because people panicked and they were thinking about worst case scenarios. But by looking at the situation, analyzing what was really going on, we were able to find out ways where we could minimize the transmission of virus from human to deer. And we also were able to assure the deer farmers and the public that there was really no danger. And I think that was a very, very important thing.
So that's pretty much public health here, interceding with animal health, with the concept of One Health. We're talking a little bit about the Survey Health Research Initiative. What is your role with this group?
Well, as a virologist, I initially got involved in this manner. The biggest concerns here in Florida were arthropod-borne viruses, arthropod being, in this case, insects, biting midges, because they transmit the viruses that cause two diseases that can be fatal in deer, blue tongue virus and epizootic hemorrhagic disease virus. And the outcomes in deer that succumb to these viruses is horrible. They start bleeding from the nose and they have bleeding in their internal organs. Those are very, very devastating diseases if you have outbreaks in your farm. And we were seeing this. Some of the farms had die-offs due to these viruses. Now, it turns out we weren't sure which particular strains, so that would be like versions of the viruses, were in circulation. And the reason that's important is it's possible to make vaccines to particular strains. You can think of influenza virus. Every year you have to have a new vaccine because the virus mutates. So if you have vaccines that work against particular strains, you're able to protect the deer. What we found in Florida was there were many strains of these two viruses, many more than anyone knew. The other thing is these viruses have what we call split genomes, segmented genomes. That means you can kind of think of like humans have chromosomes, 23. So with these viruses, they have 10 segmented genomes, and when two different strains infect an animal, those segments can mix and match. And when they do that, they give rise to new variants of the virus. And so you might be resistant to one or the other, but when they mix and match, they come up with new virus strains that you may not have resistance to. And for the cherry work we've been doing, we've been finding that these viruses are mixing and matching to a greater extent than anybody knew. And so that complicates the approaches you use to putting an end to outbreaks. For example, if you have vaccines, which vaccines should you use? Should you use a combination of many of them or just a few of them? So we're making a lot of progress there because we have already shown that in some of the farms, vaccines have been very, very effective. Now here's the catch. Because we verify what's in the animals, we have found additional viruses that no one knew even existed in Florida. And we have found some brand new ones as well. So you can imagine the difficulty for a veterinarian. Animals can't talk. So when a veterinarian looks at an animal that doesn't seem to be doing too well, the veterinarian can have all the knowledge in the world, but they may not be able to precisely tell what's killing or making the animal sick. And that's because a lot of viruses cause very similar illnesses. They're totally different viruses. And if you're immune to one, you won't be immune to the other one. But it complicates things because it's not the same virus. The vaccines won't work. And the way to stop the transmission may differ. So in this particular work, for example, we found a pox virus that no one knew existed in Florida. It's a mule deer pox virus. Whereas in Florida, we have white-tailed deer. No one knew that that particular virus was here. We found something called Mobuck virus, which was first described in Missouri, M-O-Buck. That's what it stands for, Missouri buck. But we found it here in Florida, and makes you wonder why or how it got here. We have now found additional viruses that are very similar to the ones that cause blue tongue and epizootic hemorrhagic disease virus. They're very similar when you look at their physical properties and their genetic properties, but they're entirely different viruses. And we're not quite sure what exactly they do now. We've now found 5 different ones. So we've aptly referred to them as cherry orbiviruses. And this is one of the things I want to look at moving forward, trying to understand why no one previously described those viruses. And we don't know if they're brand new viruses that are only found in Florida. But the point is, we're finding them. We're also finding that the extent that these viruses exist in Florida is larger than what anyone guessed before, like pox virus. Now we know it's found throughout Florida. We thought maybe it was found just in one farm. And so by finding out more about these particular viruses, we can better protect not only the farmed animals, but also the wild ones. And one way to do that is by also controlling the insects that spread those viruses, the blood-sucking insects. We're also making a lot of progress there. We're finding out which insects spread some of these viruses and then how to control those. So this is actually a very complicated and very scientifically rich project, but it's really giving us a lot of new avenues for research. So this is really helpful not only for deer, but I remind people a lot of these deer farms are near cattle farms, and thinking about the One Health concept, we might be seeing pathogens that are shared with cows. So we don't know what these viruses might be doing in cows as well as other animals. And we keep finding new viruses. My big concern is one of these are also going to affect humans. We don't know that yet. But we've recently found a brand new virus that's never been described in the U.S. before. And it's very different from viruses of the group. And I have no idea if it affects humans, no idea whatsoever. So those are things we're following up on.
So in other words, a lot of viruses around. I think one of the things that always comes to mind when we think about viruses directly associated to the disease. So are all those viruses we're encountering highly pathogenic? Are all them affecting the deer or affecting other livestock greatly?
That's a very, very good question. And it turns out we're all, if you take humans alone, we're all infected with viruses. I don't care who you are. If I check your saliva, I'm going to find the thing that causes kissing disease, Epstein-Barr virus. Now, if you're healthy, your immune system keeps it in check. But it's been associated with cancers, for example, depending on your stress levels. We once tested graduate students right before exam. The amount of virus in their saliva was much higher. And then as they come down after the exam, the amount of virus went down. So that's stress. So when you farm animals, stress is very important, right? That's one of the things we have to take into account. If you put deer in a pen, are they stressed? If you put cows in a small field, if you have a cow that's not around a protective bull, are they stressed? Does that make them more susceptible to some of these viruses? Those are things we have to find out. So the question is, we're finding these viruses, and we're finding them in dead animals. We find them in the bleeding organs, that sort of thing. Are these viruses what caused the illness, what caused the pathology. Those are things we have to find out. And in biology, things are very complicated. We're very careful not to jump into conclusions because it's like I say, some of these viruses might be very dangerous, but only to newborn animals, but not to adults, and vice versa. So a lot more work needs to be done. But what's important is first you find the viruses, so you know what's in circulation. Once you know what's in circulation, then that's where the science comes in. You try to figure out what the significance of those viruses are. Are they things we need to be worried about, or are they just harmless passersby? And that's what we're trying to do in this Cherry project, really figure out what are the important pathogens and how do we control them.
Yeah, that's very impressive research work. So I'm curious, given the growing challenges farmers face today, there is an increasing need for research that can help them adapt and thrive. But what kind of research do you think is most urgently needed to tackle these threats? And how can farmers themselves play a role in supporting or conducting the scientific research?
Oh boy, I love that question. Yeah, there are three things that need to really be done. Number one is we have to catalog what the viruses are. The second thing is we have to figure out how the animals are getting the viruses. What's transmitting them? Is it a case that there are rodents that come out at night that transmit some viruses? That may be the case sometimes. Are the viruses somehow in the food they're eating? That's possible, but in our case, it's typically the bugs, the biting midges, but also mosquitoes. And how do you control them? You know, that's not so simple. Some mosquitoes are daytime biters, some are nighttime biters. The biting midges there are many different ones, and they all have different life cycles. They have different biological system preferences where they breed. Are they up in the trees? Are they down? Do they come out at night? Do they come out in the morning? Are they out when it's humid? Those are all things that are very important because the way you control each one of them is very different. For example, mosquitoes. You know, a lot of people, when dengue and Zika came out, were saying, oh, you just sleep under mosquito nets. That's great. But the mosquitoes that transmit dengue and Zika are typically daytime biters. So it doesn't matter if you have a mosquito net. So these are complexities that we have to iron out. So we control the transmission, but the last thing is how do you treat or prevent these diseases in these animals? For example, if you have good vaccination and you do it widespread enough, it's possible to do it to the extent where the virus itself becomes extinct. That'd be great. So, for example, with rabies virus, there have been efforts to do widespread vaccinations, and sometimes they do what's called baiting. They'll put specifically genetically modified rabies virus in some type of fish meal or something, and they spread this in areas where there are lots of skunks or foxes. They eat them, and they develop resistance to rabies. There was a time when we thought that we could do something called drive the virus to the sea, meaning vaccinate starting from the center of the country onwards to the East Coast and get rid of the virus that way. So there are ways of having these concentrated approaches to getting rid of pathogens, and that's the goal. Me as a virologist, my entire existence really is to find out about the virus and find a way to either effectively treat or get rid of the virus. This is going to be through vaccines or antivirals, whatever. But those are things that would be very important. And I think, by looking at what's happening with the deer, again, remember that things might be going both ways, human to deer, deer to human. But we have companion animals, dogs. What about chickens in those farms? Do we have the case where things are being spread between them? So we want to control that. We want to eradicate the viruses. And it's possible if you have funding to tackle that. And I'd like to remind people at this stage, some of the vaccines that are being made already are showing benefit for the deer farmers. And we just need to keep that up.
So you mentioned vaccines. Which vaccines are available at this point for the deer farmers?
So I don't want to pin myself down right now because a lot of them are experimental. But the vaccines are being made in collaboration with a vaccine producer in South Dakota. I can't remember if it's North South Dakota. But anyway, they're being made available on a trial basis, and the farms that have tested them so far, according to my colleague Dr. On Campos, have been showing beneficial results. The number of fawns that are dying, at least due to the viruses that the vaccines are designed to control, dramatically reduced. Now we have to figure out what all the other pathogens are and ways to get rid of them.
Not an easy challenge.
No.
So the Conservative Health Initiative has scientists in many areas, including virology and vet medicine and even agronomists and animal scientists. So you work a lot with, work across many disciplines, and it's very heavily citizen-driven science, right? You have a lot of
yes.
What's the role of the citizen science into the detection, surveillance, and also the tackling some of those viral issues?
You take a scientist like myself, who's lab-based, and you put me in the field, and if I'm not trained to look at animals very carefully and notice all the subtle things that, they do, I'm pretty useless, right? And when humans get sick, I always you have to listen to what the mothers say, because the mothers are the ones who are around the children, and they can tell you better than anybody else if there's a change in behavior, what's going on. They have the pulse, they know what's happening. So in these deer farms, it would be impossible for us to have cameras and robots watching every animal 24 hours a day. I mean, yeah, I guess it's possible, but you can imagine the expense and the time wasted and so forth and so on. It's always better to have people who are actually on the ground working with the animals, because they're the ones who really can tell you what's going on. And animals have personalities, right? So if you go to a textbook, everything's night and day. Where someone on the ground who's working with the animals can tell you the very subtle changes in behavior. And they know things. If it's breeding season, those males are going to be acting different, right? If it's a doe that's getting ready to have a fawn, she's going to be acting different. You need people, citizen scientists, people on the ground who can tell you that there's something going on the farm. They can tell you that they're noticing a difference in behavior or something. The animal's not eating. The animal has diarrhea. They are the ones who really tell you what's going on. This is something that is intertwined, and the success of what we do is totally dependent on having citizen scientists. So I like to think of this again as another component of One Health. It's everybody working together. What if the farmer says, I gave a new feed and they're not eating? Well, me in my laboratory, I wouldn't know that. But we all know, we as scientists know that nutrition is very important. You have a person or animal that's not well fed, their immune system's just not going to be working optimally, right? So everybody working together allows you to better understand the big picture. That's what you need to know, the big picture.
Let me transition, keep on the topic of citizen science and monitoring and all that. Let's transition to age 5 and 1. I know we're involved with the monitoring group. We're both involved on that. I recently interviewed Dr. Ben Anderson about the biology of the virus and ecology of the virus. We also talked to Dr. Cindy Sanders about the effect that it's having on the local fairs and the state fairs, and Colin Larson about the dairy industry. Those are previous episodes for our listeners. So what's your role into this H5N1 surveillance, and what are the challenges you are facing with this work?
Well, right now I'm just acting in an advisory role, but before I joined the University of Florida, I did lots and lots and lots of work with H5N1, and it turns out there's a strong misunderstanding about H5N1. So, for example, it's a type of highly pathogenic avian influenza virus. Now, it's not the only highly pathogenic avian influenza virus. And when you say highly pathogenic avian influenza virus, HPAIV, that's a USDA designation. So the CDC, when I was working with it, always used to point that out because it's highly pathogenic, meaning that's what it does in poultry and in some wild birds. Now, influenza viruses mutate quite a bit, and there are many different strains. You can think of them as flavors. I worked with what at that time was the most dangerous strain, and it was from Vietnam. And at least 50% of the people who were infected with that virus supposedly died. I say supposedly died because we only knew about the people who died, but there was always a possibility that the same virus was affecting a lot of people, but never got reported. Okay? So we shouldn't automatically assume that just everyone who was infected by that virus would die. Now, I worked with strains that were of moderate pathogenicity, high pathogenicity like that one, and very low pathogenicity in animal models, mammalian animal models, in this case, ferrets. So what was interesting was the viruses that were highly pathogenic in humans were also highly pathogenic in ferrets. I remind people I also worked with viruses, H5N1, that were isolated from humans, and the humans were barely sick. So just because it's H5N1 doesn't mean it's going to be highly pathogenic in humans. So we have to recognize that the effects in animals, like in birds at least, and in humans may differ. The other interesting thing is we knew way back when that cows could get infected with the virus. Now, the word infected is tricky because in virology, you can be infected by a virus and not have an illness. Like I said earlier, Epstein-Barr virus, everybody has viruses in them, whether they get sick or not. So this is sort of like when Zika virus came out, only 1 in 10 people had infections with Zika virus, got a rash, fever, illness. The rest didn't even know they had it, and they were able to transmit it sexually, not knowing the virus had affected them, that sort of thing. So infection, most people understand that from when they go to their physician and they have an illness, the physician will say, you have an infection, right? But we have to be very clear what that means. So with these avian influenza viruses, we knew that cows, this was in around year 2009, we knew cows could get infected with the virus. Didn't do very much in the cows in animal tests that were done. And so it's misleading when the media tells you, oh, the virus is now mutated and it's affecting cows for the first time. Now, that's not really true. Okay. You see, people panicked because they knew that in the past, some of the very dangerous strains had induced a high mortality rate, supposedly 50% or higher in humans. And so they were afraid that what showed up in cows would do the same thing. But so far, the viruses that have shown up in cows in the U.S. have not had or induce that level of highly virulent illness in humans. And when I say that, the caveat is in people who are relatively healthy, because if you have all kinds of underlying illnesses, almost anything can cause a lethal illness, right? So we shouldn't panic. What we have to stay on top of is this. Number one, the H5N1s that are in circulation now haven't mutated to a form that's easily transmissible person to person. We have to make sure that doesn't happen, right? So that's a big concern. And the other concern is people have looked at what happens in cats. They say, oh, the cats are getting a brain disease and we're worried that's going to happen. Well, I can tell you when I looked at the very dangerous strains from Vietnam and did ferret studies, those strains always affected the brain. And we always found higher amounts of the virus in the brain than in the lungs. So we have to be very careful once again, because there was misunderstanding. People say, oh, that's influenza virus, that's a respiratory virus. We only look at what's going on in the lungs. Well, you have to do the big study, and you may be surprised what's going on in other parts of the body other than the lungs, right? Because what happens a lot is, even with what happened in the early 2000s, people in Vietnam, Indonesia would get sick, and there would be x-rays. It would just be of what was going on in the lungs. No one was paying attention to what was going on in the brains. So, yeah, we have to be concerned. We want to make sure that the H5N1s don't mutate to a form that's easily transmissible person to person, and we have to make sure they're not neurotropic and affect the brains, because that would be a very bad thing if that happened.
Yeah, so building on all that, we've seen how H5N1 can spread across different species, from wild birds to farm animals, even raising concern about potential spillover to humans. With that in mind, what measures can be taken to reduce the risk of H5N1 transmission in both farm and wild animals?
That's a really good question again. And the thing is, we have to understand how it's transmitted. I'm going to tell you this story. When COVID first broke out, my lab was one of the first to show that the virus could be infectious if it was in the air, right? Airborne transmission. Everybody said we were wrong. Now we recognize that that's the main way it gets transmitted. So let's look at the H5N1. How exactly are the cows getting it? Well, we like to blame birds, but supposedly some of the cows that have gotten sick were inside barns without birds. So how'd they get it? And there are many things we don't quite fully understand. We know that cows are sometimes fed bedding from poultry, bedding being the straw or hay, that the birds poop on. And the idea is that material is high in nitrogen and very nutritious for cows. And maybe they get it from ingestion of that material. Now, a lot of people say, no, you can't get the virus that way because it won't survive acidity. And that's a lot of speculation, because birds, when was the last time you saw a bird coughing or sneezing? Can you remember? You haven't?
We haven't.
Oh, my gosh. Well, that's because they really don't do that. They transmit the virus to each other, oftentimes by oral fecal routes, right? So ducks with the virus will go into a pond and they excrete a lot of that virus into the water in their feces. But it turns out the feather follicles also can have a lot of virus. And that was shown years ago by someone who actually worked for the USDA. And what he did was he got chickens that were infected with H5N1, and he removed their feathers like they do in wet markets if you go to Asia, where they clean the animal for you. And there was enough virus through that plucking action that got into the air that was able to then cause infections in chickens about 21 feet away. So the point is, we can transmit the virus using artificial means. We have to make sure we're not creating aerosols, through fans or maybe just wind power or something like that might transmit the virus, right? It's important to understand that, because it's possible to put maybe air filters in some of these poultry operations or barns where there's concern that virus from the outside is coming in, right? That's one. People thought about that sort of thing. But we also have to consider what we're feeding the animals. Now, cats in these barns supposedly got the virus by taking some milk, spilling when they were milking the cows. Well, in the early 2000s, when the H5N1 outbreak happened in Asia, there were reports that people were getting infected by eating raw duck blood. So that's something that some people in Asia do. And you can look at the web and you'll see pictures of raw duck blood in soup bowls. And they dress it up with scallions on top and maybe some peppers, that sort of thing. The point being that ingestion is a way that some animals can get infected. And humans are animals also. So the notion that stomach acidity kills the virus is not quite correct. And makes you wonder about consuming raw milk also. Can you get the virus that way? Possibly. And you can get other things through bacterial infections, right? So one just has to use common sense and take precautions. Like I say, luckily the virus, the H5N1 right now, is not causing widespread lethal disease in humans. But we have to stay on top of that, because these viruses mutate very quickly, and they may mix and match their genes, because they also have segmented genes, with some other influenza viruses that are in circulation. So we need to stay on top of that, because something bad might emerge, and that's something that we really need to pay attention to.
So you mentioned the oral fecal route. I think it's very interesting. A lot of our especially small producers here, they have mixed livestock systems, and they have sometimes cattle or small ruminants, and also running birds. And more often than not, they are drinking or feeding from the same troughs. That would be a high risk of exposure, right?
Yes.
One question that was asked me this weekend in a field day, and I was not sure how to answer, because I brought up the issues of the cross-contamination between species, especially when you have ruminants and poultry. And this farmer asked me if I'm running, in the case was sheep, and I'm also running poultry, chickens in my farm, a pasture-raised chicken. Which one should come first? Do we offer that pasture for the chickens first, or for the grazing animals first? And how long we need to wait between the two? And I was kind of lost in an answer. My answer was we're probably going to, since it's an avian influenza we're looking for, we're probably going to use those birds after we graze the ruminants.
That's a really, really tough one to tackle. And the reason is there are all these wild birds who are always entering into the process, and influenza viruses are carried by wild birds, and they can be found in poultry also. So how do you keep those wild birds out of the pasture? Ducks and waterfowl can come in and utilize ponds. And it turns out, especially in cold months, they've seen this in Alaska, these influenza viruses are shed into the water, and they can even overwinter, survive the winter, depending on how cold the water is, get into the mud, and so forth and so on. So the biosecurity is the big issue. And I know with poultry, a lot of people want pasture-raised animals, free-roaming animals, that sort of thing. Will it ever be entirely safe to keep them around the other animals? Probably not. So, should we vaccinate them? They do in other countries, and that might be a good idea. A vaccination isn't widespread yet in this country for H5N1, but in Asia, they do it, and a lot of people are afraid about what the costs might be. But there are vaccines that aren't that expensive. That might be one thing to look at. There's a lot more work that needs to be done here, because is it going to be the case also that the different H5N1s are going to require different vaccines? Maybe. So we know the virus in California that's broken out in cattle is not quite the same as the one that dominates in Texas. And then if you go to the upper Midwest, ours is a little different. And then in Washington State, a former colleague from the University of Florida, Tom Walczak, who's now working in Pullman, Washington, he found the H5N1s that were affecting different species up there was different. So these are all different genetic lineages. And the ones that predated all these a few years ago caused deaths in Florida. So in Florida, it's about 3 or 4 years ago, We had a lot of buzzards and waterfowl died of H5N1. Genetically, and the virus disappeared. I don't know why it disappeared other than maybe the resistant animals survived it and it got extinct. But the virus that broke out in Florida is, again, distinguishable genetically from the other ones. So, we just have to find out which particular variant is in the area. Luckily, to my knowledge, Florida is not really seeing outbreaks this time. Maybe we're going to be safer than other ones because maybe the animals that developed resistance are what are predominating here in Florida. But I don't know. We just have to stay on top of it.
And again, that's importance of your work with the surveillance, monitoring the evolution of those viruses. Dr. Lednicky, appreciate your coming to our podcast today. Miranda, appreciate co-hosting. And for those that want more information, we're going to put some links into the description of this podcast for both the work that we're developing with the Survey Health Initiative and also with the monitoring with the avian flu H5N1 here in Florida. Thank you so much, and I hope to have you here in future for us to talk more about those viruses and vaccines and protecting our livestock. Thank you.
Thank you.
Cow Talk Podcast was brought to you by the University of Florida Forage Team with technical support from the University of Florida College of Agriculture and Life Sciences Faculty Instructional Resource and Support Team. This episode was produced by Marcelo Valal, Olivia Maule, and Ashley Kanabrowski.